Good evening, everybody. Good evening, all the students, all the subscribers of Biotechnica. Welcome to this Brahmastra Series. So, as usual, let's have some quick hellos around just to make sure that you people are able to hear me. Good evening, everyone. Good evening, Komal, Karen. Hello, everyone. Come on, can I have some responses from all you people? Not audible. Yeah, I should be audible. Uh, can you hear me, everyone? Am I audible, everybody? Please let me know. Audible, right? Okay. Komal, if you're not able to hear me, please rejoin. Okay. All right. Now you can. Okay, great, Komal. Okay, so today, as you know, we are going to discuss about translation mechanism, that is from unit 3, that is molecular biology or the fundamental processes, as the name of the unit go. Now, unit 3A and 3B comprises of DNA replication, then it comprises of transcription, and C is translation. So we'll be talking about questions regarding this translation mechanism. Although, because translation is an in-depth process and there are a lot of questions which over the years has come, so I have divided the translation, the questions from translation we'll be dividing into two sessions. Okay, so one session today and one session maybe in the next week we will have, where we will discuss the questions from translation. So in today's session, basically we'll be covering the initial segments of translation. Okay, so in that we will combine the initial requirements of translation. Okay, so quickly, can we have what all things do we require for translation? Components for translation. So what all components do we need? First, what do we need? Quickly, you people tell me, I will write. mRNA, absolutely correct. Then ribosomes, right? Because ribosomes is the organelle where the translation will happen. Okay. Now, the information which is there in the mRNA is in the form of genetic code, right? So we also need to have an understanding of the genetic code. Then we need a translator molecule or an adapter molecule which can convert this genetic code into the meaning of amino acids. So that work is done by transfer RNA. Okay, yes, absolutely correct, Achal. Now, this transfer RNA, what it is? So, transfer RNA is a molecule which will have a sequence of nucleotide that is called as anticodon, which will pair with mRNA, and at the same time, tRNA will also carry an amino acid. Now, the process or by which tRNA is joined with an amino acid is called as charging, and for that we require an enzyme called as amino acyl it's an enzyme, amino acyl tRNA synthetase. Okay, so once we have all these components, right, then the translation will be starting. Okay, so today whatever questions I have taken, so they will comprise of this segments. Okay, I have not taken any question from the actual mechanism or translation, that we will cover in the maybe in the next session. Okay, but in today's session we'll talk about the questions from this initial part. Okay, all of you ready? So I have tried to take most of the questions from previous year CSI papers only, and maybe some questions are there which are equivalent to that label, but it is taken from some other source. Okay, yes, definitely. Yeah, Surendra, I did CS easy. I did not write amino acids. Yeah, definitely you need amino acids. Without the amino acids, definitely the polymer of amino acid, which is protein, cannot be made. So starting with question one, which part of ribosome identifies the Shine-Dalgarno sequence? Okay, so before we answer this, can we, yeah. So first of all, tell me what is Shine-Dalgarno sequence, or yeah, what is Shine-Dalgarno sequence, and where is it present? Where is the Shine-Dalgarno sequence present? Yes, where is Shine-Dalgarno sequence present? Absolutely correct, Mayank. So, Shine Dalgarno sequence is present on the 5 prime side of mRNA. Okay. So, if we have an mRNA like this, so it is the 5 prime end. So, near the 5 prime end only, we have this SD sequence. Okay. Now, this SD sequence, which is present on mRNA, so that will pair with what? It is going to pair with the 16S rRNA of small ribosomal subunit. Remember, when we talk about the ribosomal subunits, right? So ribosome is always made up of a large subunit and a small subunit. The large subunit carries the peptidyl transferase center, okay, which will catalyze the formation of peptide bond, whereas small ribosomal segment, it carries the decoding center. Okay, now this decoding center means what? It is the site where the tRNA will bind with the mRNA to decode it. Now, if mRNA and tRNA's binding is happening there, okay, that means what? The mRNA is attached to the smaller subunit. Now, how the mRNA attaches? So that attaches, so or how the ribosome will know where exactly the initiation codon is, because any random sequence from any random nucleotide, the translation cannot start. Translation can only start from the initiation codon, right? So initiation codon has to be placed properly of the mRNA within the ribosomal segments, right? So that is done by interaction of SD sequence, that is Shine Dalgarno sequence, which is present near the 5 prime end of the mRNA, with that of the 16S rRNA, which is a component of the small ribosomal subunit. So basically, the image looks like this. Okay, so where you have the 16S rRNA, okay, you see this is the 16S rRNA, which is part of the small ribosomal subunit. So this green one is the mRNA. So mRNA Shine Dalgarno sequence. Okay, this is Shine Dalgarno sequence. So the Shine Dalgarno sequence will pair with the 16S rRNA sequence, and that will do what? That will make sure that the initiation AUG, the initiation codon, is placed correctly at the P site. Okay, from where the translation will start. Yes, absolutely. That's correct, Karen. That the Shine-Delgado sequence, it's a conserved sequence, and it has been seen that in the organisms where there is mutation that changes the Shine-Delgado sequence a lot. So in them, the series, the efficiency of translation reduces. Whereas in those mRNAs where the Shine-Delgado sequence is close to the conserved sequence, okay, more is it similar to the conserved sequence, then then the translation becomes more stronger. Okay, is it understood? So answer will be 16 srRNA. Now next question. What would be the tripeptide produced by translation of a transcript produced by the following DNA sequence? Okay, so now this was a CSIR question, and I think it was just 2 years back only this question was asked. So here there is a tripeptide. So you have to find out what tripeptide sequence will be produced when the DNA sequence is given. Okay, now in this case you have to understand and remember. One second. Yes. Okay, so yeah, I will give you a couple of minutes. Tell me what will be the tripeptide produced by this sequence. Come on, only three people has answered. Okay, yes. So let's have a look here, guys. Okay, so the sequence that is given is in DNA. Okay, so the DNA sequence, directly looking at the DNA sequence, for sure he will be not able to find out like which codons are present. And if you want to predict what tripeptide sequence is produced, then you at least have to have the idea about what codons are present, right? So this is the DNA sequence. So immediately you have to convert it into the mRNA sequence. So what will be the mRNA sequence? So for sure, this will be the 5 prime end. Okay. Then it will be A will pair with U, U, C. Okay. Then A, U, G. Then A, G, A. This is 3 prime. Okay, please forgive my writing. It's becoming coming very bad here. Not sure. Okay, 1 second. So mRNA sequence is coming as Okay, I hope all of you can make out. So it is coming as UUC, AUG, and AGA. Yes, or mostly people have already given the correct answer. Yes. Okay. Now, so this is the mRNA sequence. This is in the mRNA. I am really not sure why I'm not able to write here. So this is the mRNA sequence anyways. Okay, so from here, so often you all ask me in the class that, ma'am, do we have to remember the entire dictionary of genetic code? So I always tell you that you don't have to remember the entire dictionary. What all you have to remember is the stop codons, the three stop codons, the initiation codons, and also the codons whose meaning is different in cytoplasmic genetic code and in mitochondrial genetic code. Okay, now I'll tell you that with that much information also you'll be able to crack it. Okay, so you know AUG, right? AUG is the initiation codon. So at the middle there is AUG. So for sure AUG will code for what? Methionine. Does not matter whether AUG is present in the middle or in the beginning. AUG will always code for methionine. Methionine can be coded by a single codon. AUG is the single codon that codes for methionine. Okay, so if it is AUG, that means in the middle it has to be methionine. Okay, now here comes the fun of multiple choice questions. So now you check the options. Does this have methionine here? No. Does this have methionine? No. Does this one has methionine? No. Okay, so there is only one option that has got methionine, so that is correct. Okay, now again I am telling you that CSIR never wants to check your memorizing ability. Okay, they don't want to check how good you are in cramming things. They want to check how good is your basic understanding. Okay, so if you have this as the DNA sequence, right? So you have to understand that first thing you have to do is convert it into mRNA sequence. Now when you are doing it, T cannot be there in mRNA, it has to be replaced with U. Okay, so all these basic concepts when you have, and when you remember the initiation codon is AUG, which will always code for methionine because methionine do not have any other codon. So all these things will come clear to you. Okay, so then you see they did not create any further confusion. There is no two options with methionine in the center, which they could have done very easily, right? So what does that mean? That they don't want to check how good you are in remembering things. They want you to check how good are you in understanding the concept. Okay, now methionine, yes, you can find out. Now at the same time, AGA codes for arginine, we know. How do we know? Because when we talk about the differences in the meaning of codons in mitochondria and cytoplasm, that time we know that AGA in cytoplasm codes for arginine, whereas AGA is a stop codon in mitochondria. Do you all remember? So from there also we know that arginine codes. So arginine can be coded by AGA. AGA is one of the codon for arginine. All right. So answer will be option D. Is it understood, everyone? Komal, I think you only asked me in the class, right? Was that you, Komal, who asked me how to answer this question? Okay, is clear? All right. So moving on. Yeah. So before that, a quick announcement as usual for our Watson Junoon scholarship. It is the enrollments are still on, and you can please call our toll free number given here. for more information and any update or any question that you have regarding this Watson Junoon scholarship. Okay, next question. Apart from 20 naturally occurring amino acids, 720 unnatural amino acids have been reported in the literature. If all the 720 were to be accommodated in the genetic code, what is the minimum number of base needed in a codon? Yes, I want you to answer also. If all 720 were to be accommodated in the genetic code, what is the minimum number of bases needed in the codon? Okay, so to answer this question, we have to understand how it was found out that codons comprises of three nucleotides. We know, we always say, right, what is codon? Codon is a group of, or it's a triplet of nucleotides. So if a codon comprises of three nucleotides, so how did the researchers come to that conclusion? Okay, so what did they see? That there are 20 naturally occurring amino acids. So if we have to code 20 naturally occurring amino acids, so if we have to code 20 naturally occurring amino acids, okay, with four base pairs. So four different base pairs are present. I am very sorry, guys. Just give me 2 minutes, okay? Let me restart the PPT. I'm not sure why I'm not able to write at all. Just give me 2 minutes. Okay. So I was telling you that to understand, to find out the answer of this question, we have to first understand how the how is it done. Okay. So how did we find out that codon comprises of three nucleotide? Okay. So the idea is, if there are 20 amino acids, so definitely at least 20 unique codons are required, right? 20 unique combinations are required. So at least 20 unique combinations are required. Okay. So if there are only, so base pairs, four different base pairs only. So if they are coming in group of two, okay. So if they are coming alone, only one base pair, then maximum four combinations can be there. Okay. Which is less than 20, not possible. If they are coming in group of two, okay, then 4 to the power 2, that many unique combinations only are possible, which will be 60. Okay, again less than 20, so it is not possible. But when they are coming in group of 3, that is the only time when it is possible that 4 to the power 3, so that many unique combinations can be there. So 4 to the power 3 becomes equal to 64. So that is how the scientists, they found out that in a codon there are three nucleotides which are present together. Okay, because then 64 is greater than 20, so at least 20 unique combinations are possible here. Okay. Now what you have to do, so you know that there are four base pairs only available. So in how many base pairs should come in combination to form a codon, so that you will get at least how many unique combinations? 720 unique combinations, right? So in how many codons, how many nucleotides should come in a group, so that we will get at least 720 different combinations. Okay. So if four codons are sorry, if four nucleotides are coming together in a group, okay, so in that case, how many combinations are possible? 4 to the power 4. If you do it, you will find 256. Okay. Now this 256 is also not possible, because that is less than 720. Now how about if we go ahead and find out five combinations on nucleotides coming in group of 5? Okay, then it will be 4. I am sorry, I'm again facing the same problem. All right, I think all of you can hear me now. So if they are coming in group of 5, okay, then how many combinations are possible? 4 to the power 5, and 4 to the power 5 will be equal to 1026, which is greater than 720. Okay, so if we have to code for 720 different amino acids, then four bases has to come together in group of 5, then only it will be possible. Okay, moving on. Next question. Which one of the following statement is not correct? And you see, this is a CSIR question. Okay, this is a part B CSIR question, but you can see how easy it is. Which of the statement is not correct? Together with proteins, rRNA provide a site for polypeptide synthesis. That is absolutely correct, right? rRNA and protein, that is what makes ribosome. So this is correct. B, all DNA molecules are unbranched polymers of nucleotide. That is also true. DNA is a polymer of nucleotide, and it is unbranched. It will always be unbranched only. Option 3, DNA is synthesized in 5' 3' direction, while RNA synthesis occurs in 3' 5' direction. Now that is absolutely wrong, because whether it is DNA or RNA, when we are talking about formation of the nucleic acid or the polymer of nucleic acid, so there phosphodiester bond has to be formed. And whenever phosphodiester bond has to be formed, then the incoming nucleotides 5' phosphate attacks the 3' OH of the last nucleotide in the chain. Okay, and that is how the phosphodiester bond is formed. So it does not matter whether we are talking about synthesis of DNA or RNA, so it will be always be done in 5' to 3' direction only. Okay, and moreover, if you see option 4, tRNA anticodon may pair with more than one codon. That is true, because in tRNA there can be Wobble pairing between the anticodon of tRNA and the codon of mRNA, and because of that, which nucleotide is occupying the first position in anticodon in tRNA, that decides which all how many different mRNAs it can identify. Okay, so answer will be option 3. Is it clear, everybody? So this is a very easy question. I think all of you already understood that. Okay, next question. Imagine the RNA sequence is translated in the mammalian cytosol and mitochondria. So the RNA sequence is given to us. Following are some putative peptide sequences. Okay, putative means possible. These are the possible peptide sequences. So if the sequence, this same RNA sequence, if it is synthesized in cytosol, these are the two combinations possible. And if it is synthesized in mitochondria, then these are the two combinations possible. Okay, now find out the correct combination of peptide made in the cytosol and mitochondria. Okay, so I would like to apologize to everybody, all the subscribers here. I could not turn my video on because somehow that is making my system slow, and there is a hanging problem which is happening today. So I'll definitely try to correct it before the next session, but I hope all of you enjoyed and find this session as fruitful as ever. Okay, so what will be the correct combination, right? Okay, so in that case, cytosol is very easy, right? What will be the meaning in cytosol? That is easy. How is it easy? So what do we have here? So AUG is the start codon, so we know that it will code for methionine. AUA, even if you don't know, okay. CUG also, if you don't know, okay. But UGA, now what is UGA? Is a stop codon, right? I told you. So stop codons, initiation codons, these are some of the codons that you should know. So UGA is a cytosolic stop codon, right? So in that case, what will happen? So the termination of translation will happen, and UGA do not code for any amino acid. So in cytosol, when this RNA has to be translated, so the polypeptide chain will be only 3 amino acid long, right? Okay, so it will be this. So only option A in cytosol, you have option A, so that one only has the 3 amino acids. So that is how long the sequence of amino acid will be when it is present in the cytoplasm. Okay, now the same sequence, if it has to be translated in mitochondria, then what will be the response? Okay, now in mitochondria, what happens is, I'll just erase this. AUG is a stop codon. Okay, but now you have to remember, think about the differences. UGA, is UGA a stop codon in mitochondria? Is UGA a stop codon? No. In mitochondria, UGA is not a stop codon and it codes for tryptophan. So in place of UGA, it will not get terminated and tryptophan will come. Okay, now if you see here, so both of the sequence that we have at the fourth position has tryptophan. Okay, now if we go ahead, what do we see? AGG comes. Now AGG codes for arginine in cytoplasm, but AGG is a stop codon in mammalian mitochondria, right? So arginine will not be there, and before this only the synthesis will stop, and the polypeptide chain, when this mRNA is translated in the mitochondria, will be 1, 2, 3, 4, 5 amino acid long. So answer will be option. So in mitochondria, the sequence we will get is 2. So which are the correct combinations? So if the sequence is translated in cytosol, then we will get A. If it is translated in mitochondria, then we will get 2. Okay, so this table I have shown you in the class also, and all the people who have joined, who are not who are not enrolled for our classes, so you can use this table. Okay, this is the table that combines the genetic code, or that actually highlights the differences in the genetic code which is there in mitochondria and the one which is in cytoplasm. Okay, moving on to next question. The following diagram shows the normal sequence of the coding region of an mRNA along with the mutant version of the same mRNA. How many amino acid long will the normal and the mutant polypeptide be? Yes, I want all of you to try and tell me. Yes, what do you guys think? What will be the answer? I have not got any response from anyone yet. Come on, it is not that difficult. Okay, that's right, Lisa. Okay, so the normal is there and then there is a mutant. Okay, so how many amino acid long will the normal and the mutant polypeptide be? So to do that, you have to have the idea about how the translation happens. Okay, so the translation will always start with the initiation codon. which is AUG. And whenever there is a stop codon, so what are the stop codons? UAA, UGA, and UAG. So any of these codons come, that it will be stopped. The translation will stop. Okay. And initiation codon will always be AUG. Okay. So in the normal one, you see what? That AUG is the first codon only. And AUG is not just for show, it codes for an amino acid, which is methionine. So in case of prokaryote, it is formal methionine that is added. In case of eukaryote, it will be the regular methionine only, which is okay. So here one amino acid for that. Next group of three, second amino acid. Third, fourth, five, six, seven, eight, nine. Okay. Why only 9? Because UAG is a stop codon. And when it is a stop codon, it does not code for any amino acid. So there won't be any amino acid coming at that place. So the normal sequence will code for 9 amino acid long polypeptide chain. Okay. Now what about the mutant? Okay. So in case of mutant, what do we have? Yes, Ramdas, Hemlata, Lisa, Mayank. Yes, you guys are correct. But apart from that, many people have given wrong answer also. So check the mutant. Okay. So now in the mutant, will the translation start from first one only? No, because a mutation has happened, because of which what is the scenario, that this U has been replaced with G. Okay. And because of that, now it is not AUG anymore. So translation cannot start at that. So what will the ribosome do? It will scan the mRNA and find the first AUG codon. Okay. So first AUG codon is coming here. So translation will start from this place. So this will be the first amino acid. Then 2, then 3, and 4, and again we have a stop codon. Okay. So the mutant mRNA will code for how many amino acid? It will be 4 amino acid long. Okay. So answer will be option A. Right. Now all of you are giving the right answer. Okay. Translation cannot start without initiation codon, without start codon, Sunshine. Okay, next question. Which one of the following statement about the recognition of tRNA by its cognate amino acid tRNA synthetase is correct? Okay. So amino acid tRNA synthetase recognize their cognate tRNA by the exclusive recognition of their anticodons. Amino acid tRNA synthetase recognize their cognate tRNA by recognition of their anticodons in some tRNAs only. Amino acid tRNA synthetase cannot amino acylate a tRNA that lacks the conserved modifications in the loop. And amino acid tRNA synthetase cannot amino acylate a tRNA that lacks conserved modification in the DHU loop. Okay. See here. So how does the amino? So we know that amino acid tRNA synthetase is required for charging the tRNA with its cognate amino acid. Okay. Now amino acid tRNA synthetase is specific for each amino acid. Now the question is that because what happens, because of the Wubble pairing that is possible between codon and anticodon, so different tRNA, means tRNA having different anticodons, can still recognize the synonymous codons. Okay, so in that case, how will amino acid tRNA know that which all tRNAs have to be charged with that specific amino acid? Okay, so in this case only you can see that only relying on anticodon is not possible, because one tRNA can bind with two or more codons depending on what base it has at the 5' position of the anticodon. And also they all are, if they are synonymous codons, so they have to be charged with the same amino acid. Okay, so amino acid tRNA synthesis cannot rely on the anticodon alone. Okay, so along with anticodon, what it has to do, it has to check some specific conserved modifications which are spread all along the structure of tRNA. Okay, so those conserved sites are present in the acceptor arm also, in the TCIC arm also, in the anticodon arm also, and in the DHU arm also. Okay, so none of them alone again cannot work. Okay, and this way of how amino acid tRNA synthesis recognizes a particular tRNA that it has to charge is called as the second genetic code. Remember, for some of you I told this in today's class only, second genetic code. Okay, so out of all the statements given, which is the most correct one? So the most correct one is amino acid tRNA synthesis recognize their cognate tRNA by recognition of their anticodon in some tRNAs only. Okay, I'm not saying that it cannot happen with anticodon arm also. It can happen, but usually anticodon alone cannot happen. Okay, anticodon plus the conserved sequences will also be taken into consideration while recognizing a particular tRNA that has to be charged with the same amino acid. Okay, so answer here will be option 2. All right, moving on. Next question. Fidelity of protein synthesis depends to a large extent on the accuracy of amino acylation of tRNA with correct amino acid. However, given that the side chains of many amino acids are not sufficiently different, amino acid tRNA synthetases are often prone to misacylate the tRNA. One such example of misacylation is of tRNA having the anticodon for threonine by threonyl tRNA synthetase. In this context, which of the statements are being made about E. coli threonyl tRNA synthetase? So there are 5 statements are given: A, B, C, D, E. Choose the option that represents the all correct statement. Okay. See what happens. Let me tell you the basic mechanism. So in amino acid tRNA synthetase, they will have two sites. Okay. So one is the catalytic site, which will catalyze the transfer of amino acid to a particular tRNA. Okay, it's a particular tRNA, and one is a deacetylation site, which will do the proofreading. Okay, why this proofreading is required? So this proofreading is required in case a wrong amino acid is attached to a particular tRNA. Okay, now these are enzymes, right? So think of it like two pockets. Okay, so this is one pocket, which is the catalytic site where the amino acylation will happen. This is the proofreading site, okay, which will do the proofreading, and if deacetylation is required, then it will do. Okay, so in the catalytic site, the amino acid, because this is specific to the amino acid, right? So the amino acid specific to it will come. Okay, so now we all know about the lock and key model and the fit model, right? So we know, so in the catalytic site is designed in such a way that the amino acid to which this particular tRNA synthetase is specific can come and enter. Okay, can come and enter. Okay, so that the amino acylation can happen. Okay, now if you take two drastically different amino acids, okay, like you took tryptophan and you took glycine, okay, so they are so different that there cannot be any confusion. They cannot go into each other's catalytic site. Okay, but confusion can happen if the amino acids have similar side chains. For example, threonine and serine. Okay, now since threonine and serine, they have such similar side chain, so it is possible that this threonyl tRNA synthetase, the serine enters the catalytic site instead of threonine. Okay, so in that case, this amino acyl tRNA synthetase will add serine to the tRNA, which is for having the anticodon to recognize threonine. Okay, so what will happen? This is an anticodon for threonine, but it has got charged with serine. Okay, so this is a wrong amino acylation. So if that happens, okay, then the proofreading site will check it, and that the proofreading site will do what? It will deacetylate it. Okay, those serine will be removed. Okay, and again this tRNA will go to the catalytic site where it will charge with threonine. Okay, so the statements, now let's check the statements. So this amino acyl tRNA synthetase acetylates tRNA threonine equally with serine and cysteine. Okay, so this is wrong. It does the mistake, but with serine. Okay, now B, it possesses a distinct editing site that preferentially deacylates the mesacylated tRNA threonine. That is correct, which is called as the proofreading site. Okay, so statement B is correct. Now C, the editing of the mesacylated tRNA threonine occurs frequently in cis before the release of the mesacylated tRNA threonine. Okay, so cis means in the same place. Okay, so same place, here we are referring to the amino acyl tRNA synthetase. Okay, so if it is threonyl tRNA synthetase, okay, so its job is to add threonine only. So by mistake, if it has added serine, then in cis, the same molecule, okay, same amino acyl tRNA synthetase only will deacylate serine and replace it with threonine. Okay, so statement C is also correct. D says that it possesses a distinct editing site that does not discriminate between misacylated tRNA threonine and threonyl tRNA threonine. That is wrong. It discriminates because there is proofreading. Okay. Now see, all amino acyl tRNA synthetases don't have this proofreading ability. Okay. So it is present only in those amino acyl tRNA synthetases where in the cell there are amino acids with similar side chains. Okay. So those amino acyl tRNA synthetases only have the proofreading ability, not all. Now, E, the amino acylation and the editing site of the amino acyl tRNA synthetase is same. Okay, now that is also wrong. It is not same, it is different. Okay, so which will be the correct combination? B and C. Is it understood, everyone? Did you all understand the mechanism of proofreading that happens in amino acyl tRNA synthetase? Because once you understand the mechanism, you can answer this question very easily. Okay, next question. Each amino acyl tRNA synthetase is precisely able to match an amino acid with the tRNA containing the correct corresponding anticodon. Most organisms have 20 different tRNA synthetases. However, some bacteria lack the synthetase for charging the tRNA for glutamine with its cognate amino acid. How do these bacteria manage to incorporate glutamine in their proteins? Choose the correct answer. Okay, so here the answer will be option 3. Now, why option 3? See, glutamine and glutamate, they also have very similar side chain, right? One has the extra amino group, one has the extra carboxylic group. So what happens here is, in those bacteria which lack the tRNA, the glutamine tRNA synthetase, so in them, the amino acyl tRNA synthetase specific for glutamate, okay, will charge the tRNA glutamine with glutamate only. Okay, and then what will happen? An extra enzymatic step will happen, which will remove glutamate, or which will replace glutamine. Which will replace glutamate with glutamine. Okay, which will basically convert the glutamate to glutamine. Okay, so suppose this is glutamate. and this is glutamine. Okay, so if this is your tRNA, so this tRNA will be first charged with glutamate, okay, by the glutamyl tRNA synthetase, and then by a extra step, enzymatic step, this glutamate will be converted into glutamine. Okay, so this is how it happens. Okay, let's see one last question before we have to end the session. So I'm just rushing so that we can finish this at least 10 questions here. So when one isolates ribosome from bacterial lysate, apart from 70S, 50S and 30S ribosomal subunits, one also finds a small population of 100S, 130S and 150S subunits. EDTA dissociates this larger ribosomal subunits into 50 and 30, suggesting that they have both the subunits. Upon addition of cations, they reassociate into 70S. Okay, but none of the other forms could be detected. What is the reason for not obtaining a more than 70S form? So what we have here, so in this case, what it is saying, okay, so when we extract the ribosome from cell, okay, so from cell when we extract, so we find 70S, 50S, 30S, 100S, 130S, and also 150S ribosomal subunits. Okay. Now, when we take this higher ribosomal subunits and break down by treating it with EDTA, so they break down into 30S and 50S subunits. Okay. Now, when we take this subunits again and join them by adding cations, okay, they join, but maximum they form only 70S, not any other of this higher S subunits. Okay, so question is why? Why is it not able to form this more than 70S forms? Okay, so what are this more than 70S forms that you have to understand? So this more than 70S form, so see, the whole prokaryotic ribosome can be 70S. Okay, so if it is more than 70S, what does that mean? That more than one ribosomal subunit is present. Okay, and how is it possible? That is only possible in case of polysome. Now what is polysome? So polysome is this structure, right, where you have same mRNA on that multiple ribosomal subunits have assembled. Okay, so if you isolate an mRNA like that and then extract the ribosome, so definitely it will give a sedimentation coefficient which will be more than 70S. Okay, right? So this higher, more than 70S forms, they are actually polysomes. Okay. Now, when you are giving them cation, so 30S and 50S, why they are not forming polysome again? Because for polysome formation, just bringing the subunits are not enough. You need the other components also. You need the mRNA, you need the charged tRNA or the amino acyl tRNA. Okay, along with, so, and for loading them also you need the various translation factors. So all these requirements are there, then only the polysome can be formed. Just adding cation, this will only combine to form 70S RNA. Okay, the 70S form. More than 70S form requires the other cellular components also. So that's why it will not be there. So answer will be option 3. That 100S, 130S, etc. represent polysome that cannot be reassembled de novo without the other cellular components. Okay, so answer will be option 3. Now, one last question, and this is very easy also. Again, if the basic of translation is clear to you, you should be able to answer it. So, a tRNA is chemically modified so that the amino acid bound is different than the one specified by the anticodon. Which codon in the mRNA would the tRNA recognize? The one specified by the anticodon or the one that matches the modified amino acid? So what it is saying, suppose you have an amino tRNA, okay? So it has this, okay, let me just color code it. So you have a tRNA, okay, which has this anticodon to recognize this amino acid, okay? They both are same, blue, okay? Now what you are doing, you are chemically modifying this tRNA so that it is now carrying a different amino acid. Okay, so you replace this blue and now it is carrying a green amino acid. Okay, so the anticodon on the tRNA is not matching the amino acid that it carries. Now the question is that if this tRNA goes for translation, then which codon in the mRNA would the tRNA recognize? Okay, the codon, so if this is again, so if this is our mRNA, so which codon will it recognize? The one which matches with the blue anticodon, or the one that matches with the amino acid that it has? Quickly tell me which one will it match? Will it match with blue or will it match with green? Yes, absolutely correct, Ramdas. So it will match with blue. Okay. So codon and anticodon. Okay. The matching in the ribosome has simple criteria that codon pairs up with anticodon. Okay. And because of that only, if a wrongly acylated tRNA enters the ribosome, then the amino acid entered in the polypeptide chain will be wrong. Okay, and to make sure that this kind of mistake does not happen, because ribosome cannot check anything, ribosome can only check if codon anticodon pairing is happening or not. So to make sure that this kind of mistake does not happen, that's why the work of amino acid tRNA synthetase is so very important. It has to make sure that every tRNA is charged with the proper amino acid. Okay, so which one will it bind? So that, and it will match with the anticodon. The anticodon will match with the codon in the mRNA. Okay. All right. Okay, thank you everyone. Then with this, we come to the end of the session. I'm sorry I extended the class by 6 minutes. So we'll stop the class here, and I'm extremely sorry for the technical difficulty that we faced in between. I hope that from next session it won't be there. Thank you for bearing with me and for staying till the end. Thank you, everyone, and I hope you found the question discussions helpful. In the next session, next week, whenever I come, so we will discuss some other questions from translation, which will comprise of the actual mechanism of translation. Okay, like initiation, elongation, and termination questions from that, as well as from the post translational modification. Okay, thank you, everyone, for joining. Bye bye. Have a great day and have a good weekend, but study well.